Hi, my name is Ray Deshaies. I'm a professor of biology at Caltech and an investigator of the Howard Hughes Medical Institute. Today I'm going to give you 3 talks, and they relate to how eukaryotic cells degrade proteins and why that's important, and how that may relate to therapy of human diseases, including cancer and neurodegeneration. Now, in my first talk, I'm going to give you a primer on the Ubiquitin-Proteasome system, and important for this part of the talk is the idea that protein degradation is an essential function in eukaryotic cells. And so what I've depicted here are 2 different cell types. I have a neuron and a muscle cell, and these cells are distinguished by the fact that they have a different set of proteins at steady state. That's what's referred to as their proteome. And they have a different set of proteins because they are producing different proteins and they are degrading different proteins. And so protein degradation plays an important role in sustaining the different proteomes that these cells have. And it is the proteomes that makes them different. You can see obviously from looking at them that a nerve cell has a very different morphology than a muscle cell. It also has a very different biochemistry, as well as performs a very different biological function. And at the root of those distinctions are the fact that these 2 cell types have very different proteins due to different rates of protein production and protein degradation, even though they have the same set of genes in their nucleus. Now, as I've said, you have different rates of production and degradation. This is just showing a hypothetical cell. And when you look at a cell, you might think, gee, that's a relatively static object and there's not much going on. But in fact, new proteins are constantly being made, and to make room for them, existing proteins have to be degraded. And I just have a little animation here showing proteins coming and going, and this is really what's going on in a cell. It's not static at all. Now, if you block the degradation process, what would happen is you'd upset this balance and unstable proteins would differentially accumulate. And that's shown here where the red proteins are unstable, and so they accumulate to a much higher level when all of a sudden you now upset this balance of protein synthesis and degradation by blocking degradation. Now, people always ask me, why is it that there's so much protein degradation in cells? Why can't you just use some other mechanism to control things? It seems very wasteful. And this slide, which I've cribbed from the Alberts textbook, makes what I think is a very important argument for why protein degradation is such a common feature of eukaryotic cell biology. And what this was intended to show in the textbook is why second messengers are unstable. But the same idea applies to proteins. And so what's shown here is how fast you achieve a new steady state if you have a 10-fold repression in the production of something or a 10-fold induction in the production of something. And what you can see is that the attainment of a new steady state is a direct function of the half-life. And so if you reduce the synthesis of a protein by 10-fold and it has a 200-minute half-life, you can see it would take a very long time to achieve the new steady state down here. Whereas if it has a 1-minute half-life, you achieve the new steady state very rapidly. And although it's maybe less intuitive, the same is true if you abruptly induce the production of a protein by 10-fold. And so if you want to use regulation of transcription or translation to control a biological process, and you want that to happen quickly, it's important that the protein that you're regulating in that manner be unstable so that it can achieve a new steady state quickly. And perhaps then it is not surprising that many of the key regulators in cells, including transcription factors, signal transduction proteins, cell cycle control proteins, proteins that regulate cell death and apoptosis, many of those key regulators are highly unstable proteins, which then allows for changes in gene transcription and protein synthesis to affect rapid changes in their level. Now, the way in which proteins are degraded in cells, there's multiple different mechanisms, but within the cytosol, within the cytoplasm and nucleus of cells, the key mechanism is known as the Ubiquitin-Proteasome system, okay? And this is a vast system. So in every one of the nuclei in every cell in your body, you have about 22,000 or so different genes. Well, of those 22,000, fully 1,000 of them, so about 5%, are involved in creating a ubiquitin signal, in reading the ubiquitin signal, or in erasing the ubiquitin signal. And you'll see what I mean by that in the next slides. So ubiquitin itself is a small protein, 76 amino acids, and it gets covalently attached to other proteins. And for that to happen, it has to be activated, and it's activated by an enzyme known as the E1 enzyme. And once it is attached to the E1 enzyme, it is then transferred to an E2 enzyme, which is also known as a ubiquitin conjugating protein. The E2 enzyme then collaborates with an E3 enzyme, also known as the ubiquitin ligase, to transfer ubiquitin to a substrate protein. And the way that works is the E3 grabs the E2 ubiquitin complex, and it grabs the substrate protein, and it holds the two of them in proximity so that ubiquitin transfer can occur to the substrate protein. Now, once you have ubiquitin on a protein, the reaction can stop there, and in that case, it's very much like phosphorylation. It changes the function of the protein in some way, either its association with other proteins, or its localization in the cell, or its activity. But the reaction can continue with repeated cycles of E2, E3 action, and now you can have multiple ubiquitins transferred onto the protein. And that can occur in multiple different types of configurations. I've illustrated one here, where you have a ubiquitin chain being built upon the protein, where the ubiquitin itself becomes the target for subsequent ubiquitin additions. And in addition to attaching ubiquitin, just as is the case for protein phosphorylation, there's a back reaction. And the back reaction is carried out by enzymes. They're referred to by different names. A common one is DUBS, for deubiquitinating enzyme. And they can either trim chains down to a smaller size, or they can remove single ubiquitin modifications. And it's highly analogous to protein phosphorylation, where you have kinases putting phosphate on, and you have phosphatases taking the phosphate off. Now, you can get, when you have a chain of ubiquitins, again, that can alter the function of the protein, but in addition, it can also specify degradation of the protein by a complex cellular structure known as the proteasome over here. And in that case, the protein is destroyed to its component amino acids, and the ubiquitin is recycled. Now, what determines whether you have signaling or degradation? Well, there's a set of proteins in the cell known as ubiquitin binding proteins, which I've abbreviated here as UBP. And those proteins recognize the ubiquitin modifications, and they're specific for different types of ubiquitin modifications, and they determine the final biological outcome of the ubiquitination. Now, I've just depicted down here the number of genes in the human genome that are devoted to these various functions in the pathway. And I'd like you to note a couple of things. First of all, the most abundant member of the family in terms of number of genes are these so-called E3 enzymes. So there's about 600 of those encoded in the human genome. And that makes sense because those are the enzymes that confer specificity on the system, right? Because they bind the substrate and the E2, and therefore they determine what proteins are being modified. They are also most frequently the targets of regulation, and we'll talk more about that later on in the talk. Now, in addition, there's about 300 ubiquitin binding proteins with ubiquitin binding domains. There's 15 different domains that bind ubiquitin, and they're spread out over 300 different proteins. So you can see how you can have many, many different biological outcomes of these ubiquitin modifications because you have so many of these proteins that read the modification. But again, the outcome that we'll talk about, we'll focus in this talk, is proteolysis by the proteasome. Now, this system, as befitting its size, remember 1,000 genes in the human genome, it's terribly important for human biology and physiology. And as a result, when things go wrong in this system due to mutation, it predisposes to a number of different human diseases. And here I've just sketched out some instances where there are direct causal links between the ubiquitin-proteasome system and human disease. And this is due to mutations that are known to occur in human that predispose to these diseases. So for cancer, we have mutations in several different E3 ubiquitin ligases and a mutation in a deubiquitinating enzyme that give rise to a predisposition to a whole range of different cancers, as indicated over here. In addition, neurodegenerative disease, there are mutations in ubiquitin conjugating enzymes and a ubiquitin chaperone protein that we'll hear about in part 3. And these give rise to different neurodegenerative diseases, including ALS, which is Lou Gehrig's disease, Angelman syndrome, and others. And finally, cardiovascular disease. It's known that mutations in one of the Cullin-RING ligases, which will be the subject of part 2, give rise to a disease characterized by hypertension. And this is not intended to be a comprehensive list of diseases. These are just a subset of the ones that we know, but it illustrates the importance of this system to human physiology and human health. Now, I mentioned earlier that ubiquitin becomes conjugated to proteins in a covalent linkage, and this shows the linkage right here. So ubiquitin at its C-terminus has a glycine, glycine 76, which is shown right here, and that residue forms an amide linkage with the amino group on the side chain of a lysine residue in the target protein. So here's the side chain of a lysine residue, there's the amino group right there, and this is the amide linkage that forms with the C-terminus of ubiquitin. So essentially, a protein that has ubiquitin conjugated to it now has 2 amino termini, and it's a branched polypeptide. Now, once you have ubiquitin attached, I mentioned that subsequent ubiquitins can be attached to the first ubiquitin. And I've shown here at the bottom ubiquitin, and then the numbers refer to different lysines in ubiquitin. So ubiquitin has 7 different lysines. So for example, lysine 29 and lysine 33 and so forth. And it also has an amino terminus. And all of those positions can accept a ubiquitin molecule. So you can make 8 different types of ubiquitin chain linkages, each of which has a distinct biology. Okay, this just shows some of the linkages. So I mentioned earlier you can get monoubiquitination, that's all the way on the left, and that has been shown to be important in processes like endocytosis, protein transport, and DNA repair. You can also build chains on the lysine 63 of ubiquitin, and those have been prominently associated, again, with protein transport, DNA repair. Now, for protein degradation by the proteasome, the linkages that have been shown to play a very important role are the lysine 48 and the lysine 11 linkages. And lysine 48 in particular seems to have a dedicated role for protein degradation. Now, there's further complexity here that I haven't touched on yet, and that is that there's recent evidence that you can form branched ubiquitin chains where you have a ubiquitin in the chain that has one linkage, let's say K11, and then a second linkage like K48. And that creates a branched structure. And there's now data to indicate that those branched structures also have unique biological functions, although those studies are in their very early days. So there's a great deal that needs to be done and needs to be understood. But if you think about it, adding the idea of branches really explodes the potential complexity of this system. Now, I mentioned we're going to focus on protein degradation by the proteasome. And so here is the proteasome itself. This is a very, very large protein complex. It has 30 different polypeptide subunits, and they break up into 2 separate complexes known as the 20S core, that's right here, and the 19S cap, that's right here. Now, the 20S core is a cylinder. It's an empty cylinder, and on the inside, it has 2 copies each of 3 different proteolytic active sites. That's the degradative chamber. Proteins get inserted into the 20S core and then destroyed by those 6 proteolytic active sites. Now, to get in the 20S core, normally the ends of the 20s core are sealed, and to open them up, you have to have a 19S cap that binds. And the 19S cap has several different important components. So first, it has a ring of ATPases. They're right here. And those ATPases sit up against the 20S, and they open. It's like opening the diaphragm of a camera. They open up the 20S so that proteins can be inserted inside. There are also ubiquitin receptor proteins. There's one of them shown up here at the top of the proteasome. And those bind the ubiquitin chain and hold the substrate there so that it can be degraded by the proteasome. And now, normally the proteasome would have a cap on each end, but for simplicity, I've only shown here a cap on one end. And this is an image that was graciously given to me by Dr. Andreas Martin at UC Berkeley. Now, this is going to rotate just so you can see the proteasome from different angles. And now I'm going to step through what happens when a substrate comes to the proteasome. So here we have a substrate protein in red. It's got a ubiquitin chain on it in blue of K48 linkages, and they're attached by that yellow lysine in the middle of the red polypeptide. So the first thing that's going to happen is this substrate is going to dock, and it's going to dock by binding a ubiquitin receptor on the proteasome that's known as RPN10 right here. And so here's the substrate. It's going to come in. It's going to dock. The next thing that'll happen is the substrate is going to engage by inserting its tail into the ring of ATPases. This is the ring of ATPases down here. The tail is going to move, and it's going to insert itself into that ring. And they're going to begin grabbing onto that tail and pulling it down to pull the protein into the maw of the proteasome so that it can be degraded. The next thing that's going to happen as that pulling starts is the protein is going to move downwards towards this green protein, RPN11. And RPN11 is a protease, and we'll hear about it in part 3, that removes the ubiquitin chain, because otherwise the ubiquitin chain would sterically interfere with insertion of the protein into the proteasome. And so that ubiquitin chain is going to be cut off by RPN11. It'll diffuse away so that it can be recycled for further use. And there's the cleavage event occurring. The chain dissociates, diffuses away, and now the rest of the substrate is dragged into the proteasome and destroyed. Now that we know how the protein is degraded, I'm going to focus extensively on the ubiquitin conjugation by the ubiquitin ligases. Now, I mentioned earlier that this is part of a dynamic where you have the E2 and E3 enzymes that are busily putting ubiquitin onto substrates, but in addition, you have deubiquitinating enzymes that are removing the ubiquitins. And this is a dynamic balance that plays an important role in controlling what proteins are degraded and the rates they are degraded at. However, I'm going to focus in the remainder of my talk on these E3 ubiquitin ligases. An equally long discussion can be made about the deubiquitinating enzymes themselves, but I would just like to mention that many of the regulatory concepts that I lay out for the E3 enzymes are shared by the deubiquitinating enzymes. So there's many different ways of controlling their activity as well. So I'd said earlier that there's about 600 E3 enzymes, and in the human genome, there's 2 different domains that control the vast majority of ubiquitin ligases, and those domains are the so-called HECT domain and the RING domain. So there's 28 HECT domain genes in the human genome. There's about 300 RING domains. You'll hear in part 2 that 2 of those 300 RING domain proteins are used in a combinatorial fashion to assemble another roughly 250 or so ubiquitin ligases known as Cullin-RING ligases. And so if you add everything up, you get to around 600 ubiquitin ligases. Now, these 2 classes, the HECT and the RING ubiquitin ligases, work by very different mechanisms. So they both share in common that they recruit a substrate and they recruit an E2 enzyme. Okay? But then in a HECT domain ligase, the E3 itself has a catalytic site that accepts ubiquitin from the E2 over here, and then transfers that ubiquitin to the substrate. Meanwhile, with a ring domain ligase, you bind the substrate and the E2, but then the ubiquitin is transferred directly from the E2 to the substrate without an intermediate on the E3 enzyme. Now that may sound arcane, but the important point is that in the case of the HECT domain ligases, it's the E3 that determines the linkage, because that's where the last step of the reaction is happening, on the surface of the E3. That's where the chemistry occurs. Whereas with the ring domain ligases, it's the E2 that determines the linkage, because the chemistry is occurring on the surface of the E2. And that's critical, because if you remember from earlier, the linkages end up determining the resulting biology of the ubiquitination event. Now, another thing that's important, especially when thinking about building ubiquitin chains, is that for a ubiquitin chain to be built, there must be multiple cycles of E2 recruitment to the E3 enzyme. And that's because after you transfer your first ubiquitin, you would have to recharge. So now we have an E2 here, and the E2 is uncharged. So you'd have to restore ubiquitin on that E2, and that requires E1. However, E1 and the ubiquitin ligase, the E3, they recognize the same surface on the E2, which I've denoted here as the dark blue part of the shaded oval. And so for the recharging to occur, the E2 has to come off the E3, and then a new E2 molecule from the cellular pool that's already been charged can bind, and once one of these binds, it can then transfer its ubiquitin onto the substrate, and in that manner, you could start building ubiquitin chains. Now, this just shows a little bit more detail what I've told you. And so if you have an E3 enzyme here at the top in part A, it will bind the substrate and ubiquitin charged E2 simultaneously in part B. And then the substrate lysine, this is a ring domain enzyme I'm showing you here, so the substrate lysine directly will attack the ubiquitin linkage on the E2, and you'll get ubiquitin transfers shown in part C. The E2 then has to dissociate, and a new molecule of charged E2 can bind, that's in part E, and then it will transfer its cargo, in this case, onto a lysine residue of the first ubiquitin that was attached. And now you'll have a chain of 2 ubiquitins. This can continue for multiple cycles until the substrate dissociates. So in the end, the length of the ubiquitin chain that forms on the substrate is really a function just of the relative kinetics of the different processes of how fast the substrate dissociates versus how fast the E2 binds, transfers its ubiquitin, and dissociates to allow a new E2 to come in. So the longer a substrate is bound, the more ubiquitins it can receive. It's just a very simple idea. Now, these reactions, as I mentioned earlier, are highly regulated. And there's many, many different types of regulation that can occur. I'm not going to cover them all. I'll just give you a flavor for the types of regulation that have been most highly studied. And so one of the most intensively studied forms of regulation in the ubiquitin system is via protein phosphorylation. And so we know that E3s and substrates are both regulated. So on the left, I show you an example that's prominent with the so-called SCF ubiquitin ligases. We'll hear about them in part 2, where phosphorylation of the substrate creates a signal, and that signal then allows the substrate to bind directly to the E3. And so in that case, phosphorylation is switching on the substrate. Now, there's also a role for phosphorylation in turning on the enzyme. That's seen with the APCC, which is an important cell cycle control enzyme, where phosphorylation creates a binding site for a substrate recruiting factor, here known as CDC20, bound to its substrate, allowing them to dock on the enzyme so the substrate can be ubiquitilated. And phosphorylation also has an inhibitory role, once again, with the APCC, where you phosphorylate the adapter, the orange protein here, and that phosphorylation blocks its binding to the E3 and therefore prevents the substrate from being degraded. So both positive and negative roles for phosphorylation. There's other types of regulation as well. One of my favorites was discovered by Ning Zhang at University of Washington, where he showed the way auxin signaling works in the plant. So auxin is a very important hormone in plant photomorphogenesis, and it works by binding a ubiquitin ligase known as TER1. TER1 has a set of substrates, and those substrates can't bind on their own. Here's an example of the substrate up here, because they don't have perfect complementarity with the binding site. But auxin, in effect, makes them whole. It rounds out the configuration, so now you have a nice binding of the substrate to the enzyme in the presence of auxin. And in that matter, auxin is a co-ligand that stimulates degradation. There's also pseudo-substrate inhibitors that mimic a substrate, but they don't have a lysine, so they can't be ubiquitilated, they're not degraded, so they can just clog up the enzyme. And there's also examples of allosteric regulation where a small molecule, in this case this purple box down here, that small molecule can bind an autoinhibitory part of the ligase shown here and cause it to undergo a conformational change and move out of the way to allow substrate to bind. And there's some examples of that form of regulation as well. And there's many others that I'm not going to describe here, but just give you a flavor of how complicated the regulation can be. Now, I'd like to go through examples of how different signaling systems can work together and how the ubiquitin system can work to control a complex biological output. And this example here was derived from work that my lab and a number of other labs indicated down below here did about 15 or so years ago in trying to understand how a yeast cell goes from the G1 phase of the cell cycle to the S phase of the cell cycle where it's going to replicate its DNA. Now, a key factor in replicating the DNA is an S-phase cyclin CDK complex that's going to activate the replication proteins. That's produced in G1 phase, but the minute it's produced, it's shut off. And it's shut off by binding of a CDK inhibitor that's known as SIC1 or SIC1. Now, at that same time, there's a different cyclin CDK complex, the G1 cyclin CDK, and that complex is active and it's not subject to inhibition by SIC1. In fact, quite the opposite. The G1 cyclin CDK phosphorylates the SIC1 protein. And that phosphorylation is depicted up here as these yellow spheres. And once the SIC1 is phosphorylated, the phosphates are epitopes, if you will, for an SCF ubiquitin ligase that then binds those phosphates and elaborates a ubiquitin chain on the SIC1 protein. The proteasome then does this remarkable feat of gymnastics where it can grab that ubiquitin chain and strip the SIC1 off the cyclin CDK complex without harming it at all. In fact, it releases the cyclin CDK complex in an active form and destroys the SIC1. And in this manner, phosphorylation and ubiquitination collaborate hand in hand to drive an essential transition in the cell cycle. And a very similar process occurs in human cells with a similar set of proteins. Now, another example, and this is very critical in human physiology, concerns how your tissues sense oxygen. And this is very important because if you have insufficient oxygen, two things need to happen. First of all, you need to induce the production of more red blood cells. And second of all, you need to induce the production of more capillaries, more blood vessels to supply oxygen to the tissues that are deprived of oxygen. And the way this works is there's a key transcription factor. There's actually two, I'm just showing one here, HIF1 alpha and HIF2 alpha. HIF stands for hypoxia-inducible factor. Now, these transcription factors normally are barely detectable in a typical cell that's well oxygenated. And the reason they're barely detectable is that there's an enzyme known as prolyl hydroxylase that uses molecular oxygen to hydroxylate a proline on those transcription factors, on HIF1 alpha and HIF2 alpha. That hydroxylated proline, which I've denoted here, serves as a direct ligand for a ubiquitin ligase known as the VHL ubiquitin ligase. That then transfers ubiquitin onto the hydroxylated protein, and then the hydroxylated protein is destroyed by the proteasome. Now, what happens if you have a cell in your body that's experiencing an oxygen deficit because it's too far away from a blood vessel? Well, in that cell, the oxygen concentration is very low. And it turns out that the prolyl hydroxylase enzyme has a lousy Km, a lousy Michaelis constant for oxygen. So at low oxygen, the enzyme is no longer saturated and it can no longer operate efficiently. So the proline on HIF protein is not hydroxylated. And if it's not hydroxylated, the protein now becomes stable. It rapidly piles up, it translocates to the nucleus, and it binds to the promoters of genes that are involved in production of red blood cells and in angiogenesis, which is the building of new blood vessels. And so in that manner, your body can mount an appropriate response to oxygen deprivation. Now, it's interesting to note that VHL is a very prominent tumor suppressor protein. And that's because as a tumor starts to grow in your body, once it achieves a diameter of about 1 millimeter, already the cells aren't getting enough oxygen to sustain them. And so if they lose VHL, they can now very efficiently promote the formation of blood vessels because the HIF proteins are no longer being degraded. And so people who are born missing one allele of VHL are prone to having cancer, in particular kidney cancer. Now there's other pathways. There's many pathways in the cell that are controlled by the ubiquitin system. I've just listed some of them here. And one thing I didn't draw attention to yet, but in the 2 examples I gave you, ubiquitin is used in very different ways. In the first example, activation of degradation is used to achieve signaling, right? Where you degrade the SIK1 and signaling, you go from G1 to S phase. In the second example with HIF, it's the inhibition of degradation that achieves signaling. When you have low degradation, you get hypoxic signaling via the HIF proteins. And in these pathways, it's no different. The ones in gray here, those are cases where signaling, where the degradation occurs constitutively, and signaling, for example, by Wnt, shuts off the degradation, in this case of beta-catenin, the one I've mentioned at the top of the list. The other examples, NF-kappa B and auxin, are cases where degradation activates signaling. So in the case of NF-kappa B, it's inhibited by I-kappa B. When you have signaling, you destroy I-kappa B, so NF-kappa B switches on. Now, I've focused exclusively on regulation so far, but that's really only half the story. So indeed, the ubiquitin-proteasome system plays a very important role in regulatory biology, as exemplified by the degradation of cyclins to inactivate CDK at the end of mitosis. But the ubiquitin-proteasome system also plays a very critical role in protein quality control. So you're constantly producing new proteins, as I mentioned early in my talk, and degrading existing proteins. When you produce new proteins, a certain fraction of those are bound to misfold. And they misfold either because they just don't achieve the right conformation, because they fail to find their partners if they need to assemble a complex, or because of mistakes in translation. And at some level, the entire proteome is subject to this kind of defective production. And so the ubiquitin system plays a very, very important role in cleaning up after those mistakes to prevent them from accumulating. And just as mutations in these regulatory pathways can lead to cancer, mutations in these quality control pathways can lead to neurodegenerative disease. And so these are really the 2 major important functions of the pathway that are commanding a lot of the attention of researchers in the field. Thank you for your attention. And in my next talks, I'll cover Cullin-RING ubiquitin ligases and using the ubiquitin pathway as a target for developing drugs to treat human disease. Thank you.